for the next speaker, who is, okay. So the next talk will be online. And while we are waiting, I just want to mention that we are monitoring the online questions on Zoom. So for the online attendees, if we have any question, we are monitoring during the talks, okay? We are ready. Okay, so I will introduce the last speaker of this session is Professor Christian Liland from Norwegian University of Life Sciences. So Christian is a professor from the Department of Mechanical Engineering and Technology Management. His research interests include multivariate data analysis and, of course, machine learning and deep learning. Whenever you are ready, you can start. Thank you.
Okay, thank you for your introduction. I hope you can hear me well over there. So,
well, yes.
Yeah, good. Okay, so you already gave an introduction. We're a large team behind this. I couldn't come to the conference due to other things that are overlapping. So Ole Christopher was supposed to give this talk, but now he is also not here. So I was able to step in again. So I will be talking a little bit about the Tsetlin machine, which is part of my interest, and then DNA sequence classification, which I've also been working a bit with the last few years. So to start this, I will give a little bit of introduction to DNA sequence classification first. So for those that are unfamiliar, we are looking at DNA, the deoxyribonucleic acid. So we call it the code of life. It has recipes for tens of thousands of different proteins. In the human body, you will find these in all the cells, in all the cores of the cells. So it's a quite important molecule to know about. It has a very simple alphabet, only 4 letters, which we call nucleotides or bases. These are 4 different chemical compounds that are put together in different configurations along this molecule. They're called cytosine, and that gives you a C, guanine, which gives you a G, then you have adenine, A, and thymine, T. Yeah, the names aren't that important, but the alphabet is very important. Along the DNA double helix, you will find complementary bases. So here we see that GC and AT, they pair up. So an A will always be paired with a T will always be paired with an A. The same goes for the GC and CG. There is also readable code in both directions. So if you start looking at this from the left-hand side with the black arrow here, you can see the ATGCC and so on. That's a primary strand, and that gives you one set of possible genes. And you also have the blue strand going from the right-hand side, which is the reverse complement, which could also be relevant genes, coding genes that give proteins when they are decoded. This means that there are some complications, there are some possibility of overlaps and things. We are keeping this a little bit simpler here in our analysis. There are some protein coding genes, and that's important here, but mostly there is junk. There's a very large percentage of the DNA in most organisms that does not code for anything that we know about. So mostly it's just leftovers or good old permutations that are not used for actual building blocks. Now, the organization along the genome is a bit important too. So we have something that we call codons and amino acids. So the DNA is decoded 3 and 3 bases. So if you have, for instance, CTG, then the CTG would be one of these codons. And then we code the next 3 ones and so on. That means that with this simple alphabet of 4 possible letters, we have 4 to the power of 3 equals 64 combinations of possible codons. And because of some redundancy, typically in the last of these 3 letters, then there's 20 amino acids that this will code for. So these are the building blocks of the proteins. In addition, we also have a stop codon, TGA, which says that if you get to a TGA, this is the end of anything that you're going to read, and you restart something new. So if you are looking for genes along a chromosome, this is the end of the potential gene. Only 3 out of these 20 amino acids can be start codons. So these are potential starts for a gene. They are the ATG, GTG, and TTG. Something that is special with these is that they can also appear inside the genes. And this is where some of the trouble comes when you're trying to find which are protein coding genes and which are just something that looks like a gene because of start and stop codons, but they do not code. Another thing that complicates this a little bit is that we have 3 reading frames for each of the strands or each of the directions. In this case, you see that I put some rectangles around 3 and 3 bases. So GCA would be 1 codon, TGA would be the second codon, and so on. But you can also move this 1 base, and we get a completely new reading of this string. And we can do it a 3rd time, and this would also give rise to a new possibility for finding genes along the chromosome. And of course, we could have the reverse complement where we start from the other end with the opposite letter, and we get the same thing there. So to summarize this, these are then the 6 possible reading frames: 1st, 2nd, 3rd along the primary strand, and then we have the reverse complement with 4th, 5th, and the 6th reference or reading frame. Now I put in one specific sequence here, starting GCA, TGA, and so on. And I marked in red the ones that are the stop codons, and in blue we find the start codons here. So if you look in the first reading frame, the first codon is just some kind of amino acid. That's why it's black. And then we get the red one, so that's a stop codon. So anything that has been translated up to there would stop at that point. And then we have the blue ATG, the 3rd codon on the first reference reading frame here. That's a possible start. And there's another possible start a little bit later at the GTG, and it stops at the right-hand side with the TGA. And there are possible starts here. It could be the ATG or GTG. We do not know. We have to do some chemical analysis or testing in the labs to find out which of these are actually the start of the possible gene here. And that's just the first reading frame. And then we move 1 base, and then we look for more start and stop codons. And then we come to the 3rd reading frame, and then we see, okay, we have new hits. So there could be a gene also here. So open reading frame is anything that has a start codon and a stop codon in the same reading frame. We also talk about the longest ORF. So that's the one starting the furthest away from the stop codon. So for the first reading frame here, it would be from the ATG, the blue one in the beginning, going to the TGA almost at the end here. Determining which ORFs are protein coding genes and which are not can be considered a binary classification problem. So it's either a protein coding gene or it's not. And limiting the search to only the longest ORFs, the LORFs, makes this simpler, and it doesn't matter too much for what analysis we're going to do later. Because after we have found which are the potential genes, we will probably do some kind of extra analysis to try to find the function of the genes, how they appear in the organism and what role they have in the regulation. Yeah, and again, the LORF or the longest ORF, that's from the first potential starting codon to the first stop codon here. The data that we're going to look at here are 15 bacteria. These are well-known bacteria from previous studies. We know all their genes. They're used a lot for modeling and prediction in the literature. There are thousands of organisms that we can choose between, but these are the gold standard in a way. If you look in the graphs here, these are frequencies of ORFs, so these longest ORFs. If you see the blue part here, that's the negative ORFs, or the ones that do not code, and the purple part is the ones that are actually coding genes. So you see most of the open reading frames are not giving us anything interesting. And the proportion is quite similar, with around 4 to 6% of the open reading frames actually being coding genes. It means that there's quite imbalanced data, but it's not extreme. We can cope quite well with ordinary methods. See if I can get the next one. Yeah, there it is. If you look at the distributions of these ORFs, they are also very different. So the gene ORFs, the ones on the left-hand side here, they are the ones that are coding, and we see that there's quite a few of them that are from 1,000 bases long and up to around 4,000 bases. And then there's very few going further on. The data behind this one is from just one of these bacteria. If we look at the negative ORFs, so those that are not coding, then most of them are quite short, and there are very few of them, of the longer ones, that are non-coding. So there's if you only looked at the length of these open reading frames, we could already give an indication of if they are coding or not. So it's the shorter ones that's the big problem. So now we come to the actual Tsetlin machine part. I won't present anything about the mechanics of the Tsetlin machine. I assume that you know this. But the Booleanizing here is a bit interesting, because we can do away with only 2 bits per position along the sequence. If you use the first bit to distinguish between AT and GC, and the second bit to distinguish again between AT or A and T and G and C within those, we can do with only 2 bits. So it's extremely compact coding of the sequences. The con here is that we're missing, if you're having missing bases, which will be a problem, we have to use one of the other bases here as missing also. So we have chosen then 0, 0, that's an A, to be missing and coding for an A. So that's a bit strange, but it actually works in practice. We could also do this with a 4-bit encoding, where we use just simple one-hot encoding, saying that an A is 1, 0, 0, 0, B is 0, 1, 0, 0, and so on. And the good thing here is that having a missing base is no problem, because it will have its own code. The problem is memory-wise, because we get to double the footprint, and of course it takes more time to process. The Tsetlin machine that we've chosen to use here is just a vanilla Tsetlin machine. So none of the specialties. I'm lying a little bit, because we're using integer weighting, but except for that, it's all plain. And we've chosen a couple of values for the different parts here. So the T and the S and the number of clauses. So we've set up a tiny grid search to look across these to try to find the best predictions. Yeah, there's highly varying lengths of these LORFs, as we already seen in the graphs. So we tried to test different cutoff lengths, saying that we limit to the first 1,000 bases, or 2,000 or 5,000 bases of these ORFs. And then we pad the shorter ORFs just with zeros, to get them to a common size that we can use in a Tsetlin machine. And the longest ORFs are around 8,000 bases. The shortest are actually down to 45 bases. There could be shorter ones, but we've set a cutoff there, which is common in the literature, just to not overstrain the machinery. So we've done a typical training validation test split here, where we use training and validation to find the best combination of parameters, and then we use the last 25% for testing. So what we see here is a table of the results for the Tsetlin machine. What we see here is that we get state-of-the-art performance. All the numbers that we see here are on par with the best in the business. We see that we have accuracies in these predictions of around 98 to 99 point a lot, almost 100% accuracy for the best ones. And even if we use metrics that are a bit more strict with regard to balance, we are still doing quite well here. Yeah, among the good things here is that we don't need any feature engineering. We can just Booleanize our data and put them straight in the Tsetlin machine, and it works, yeah, off the bat. And the memory footprint here is very small. Using 2 or 4-bit encoding actually gives us very similar results so far. Yeah, even with this duality with the A and the missing having the same encoding. Yeah, I have had a master student who has looked at all kinds of other machine learning methods for this. And what we see is that they are very time consuming because they need a lot of pre-processing. They need to find some way of recoding the genes to get as much information out as possible. As a benchmark against this one, we also set up a multi-layer perceptron with 64 and 32 nodes and basic ReLU activation and a little bit of dropout. And what we see is that it uses around 3 times as much memory as the Tsetlin machine, both in 2 and 4-bit encoding. And it has very similar performance to what we've seen. So it should be said that the Tsetlin machine that we've used, we haven't tuned too much. It's only within this grid. We haven't tried all the different extensions. So there's a lot that can be done further on this one. Okay, so the take-home message here is that we have taken the Tsetlin machine, into the realm of bioinformatics. We are analyzing sequences that are close to Boolean from the start. There's a very simple encoding to get this going. And what we've shown is only a first proof of concept, a single use case with minimal tuning. There's a huge amount of different problems that can be explored using this approach. We also have some future prospects. We want to do some cross-species modeling because what we've done so far has been in one species at a time. Yeah, so there are pros and cons with doing it either inside or between different species. We are hoping that we can get more performance by pooling things that are similar, but there's also so much variation in biology that it doesn't always transfer from one species to the other one. Could be different ways of setting up what is a gene and what is not. There are also different bioinformatic problems that could be handled with this. For instance, we could be looking for species identification. Yeah, there's a lot of regulation activity in the genes. So there's a lot of things that could be tested. It would also be interesting to use techniques from text classification, image analysis, and embedding to try to make a more rich model environment for this type of data. From the spring of 2024, I will have 2 master students working on different problems that will do Tsetlin machines and DNA together. So I'm hoping to push this further next year and yeah, continue collaboration with my partners and friends. So thank you for your attention. Hope you have some questions.
Thank you very much for your talk. Very interesting. And it's quite curious to see how Tsetlin machine can be applied to very different types of applications as far as you can find some form of binarization in there. So please, the first
Thank you,
question.
Christian. So it's mostly not a question, but a comment. So it seems that the threshold was too large for your results. And then if you make the threshold more reasonable, you can get even higher accuracy. So in the afternoon, after the dinner, we'll present a talk on the optimal hyperparameters. And it looks like the threshold should be like the optimal threshold should be like a square root of the half of the number of clauses. In your example, it was too high, so you never can reach it. Well, and it shouldn't be higher than the number of clauses itself. So this is just one comment, and maybe you can get even higher results if you set it optimally. Thanks.
Yeah.
Thank you. Any other question? I think there is another question.
How many how many rules did you actually set up for your Tsetlin Machine training?
So
The number of rules.
Oh, sorry, I just lost the.
Just to explain what I mean, I wonder if you could assess the gene as protein producing or non-protein producing just based on those rules. And if it is a small number of rules, probably it would be easier to do.
Yeah,
you
so
have clauses. Have you tried to do it with a smaller number of clauses in such a way that you could clearly articulate the difference between those genes?
yeah, I've also tried outside of the range that we see here when I was trying to limit and find some reasonable place to start. I haven't gone very low. I think I started around 100 or 200 clauses. But that could also be interesting to see if we get something interpretable if we go further down.
Okay, thank you.
Well, I would like to thank all the speakers of this session, and I think we are almost ready for lunch, right?
Just a small announcement. We will have a group photo session just before having lunch. If you'd kindly, maybe leave this room and go outside the building, we'll just take a group photo. We'll have lunch now, and then we will come back at 2 sharp for the next session. And I think the plan is we'll have one technical session with 2 talks, followed by coffee break and social right after. Thank you. Let's go outside for a group photo.